Funding for this program is provided by Annenberg CPB to advance excellent teaching.
In an unseen world, microbes are the masters of survival.
I would say that so far there probably hasn't been an environment investigated on Earth that we haven't found microbial life.
It is a world more diverse than the plant and animal worlds combined.
There's a huge diversity in the natural microbial world that hasn't been described, and that we have now the techniques to be able to describe those organisms.
Today, scientists are unraveling mysteries in microbial DNA, learning how microbes communicate, and discovering how these master chemists are changing our world. Three and a half billion years ago, the sun scorched the planet. Oceans boiled, and there was no free oxygen. Yet microbes inhabited Earth long before any other living things. The question is, how? Microbes have developed amazing strategies to obtain energy and nutrients. No matter where one looks, even on the rock surface of this cave, microbes are in abundance. And because of their keen ability to exploit their environment, microbes have evolved to fill just about every niche on Earth. Today, the genetic and metabolic diversity of microbes is greater than all eukaryotes combined. There are 5,000 known species, but scientists estimate that true diversity could range between 400,000 and 4 million species. But what are these microbes, and what are they doing? Scientists are devoting lifetimes to finding out.
Wherever we go, whenever we come back from a field trip, we always come back with big surprises, brand new branches of life that we never realized existed. So there are huge discoveries around the corner. Just if we look, we'll find them. Let's just walk down here. Just be careful, okay? Just stay on.
Professor Anna Louise Reisenbach is a microbial ecologist. She's in Yellowstone National Park with some of her students, studying organisms that thrive in the steamy hydrothermal springs.
That search for the unknown is one of the biggest challenges, and that's really what drives my science. It leaves me in amazing awe of nature, because there's so little we know, and it's just so complex, but it's beautifully complex. You know, it's really incredibly clever.
Reisenbach is searching for microbial clues that will help her understand the evolution of early Earth.
In Fahrenheit, it's 194.
Places like Yellowstone provide a natural laboratory for research.
These thermal features may be relics or fossils from what it was like on early Earth. And so understanding the types of processes that occur in these systems may help us understand what it was really like 3.5 billion years ago.
Prokaryotic microbes used to be grouped together in a single kingdom. But in the late 1970s, Dr. Carl Woese, using ribosomal RNA sequencing, identified a genetically distinct group of organisms. He called this new domain on the tree of life the Archaea. Archaea and bacteria often look similar. They both have outer cell membranes and a cell wall. But bacteria contain peptidoglycan, a chemical that archaeal cell walls lack. Using gene sequencing, scientists discovered that Archaea may be more closely related to eukaryotes than other bacteria. Many Archaea and some bacteria are Extremophiles, thriving in extremely salty, cold, or acidic environments. Thermophiles like it hot and require high temperatures to survive.
And the one thing that's really amazing about Yellowstone is that all the different colors of the different pools do reflect huge chemical and temperature gradients. You know, and so that whole range of pHs and the whole range of temperatures provides all these different environments for a huge diversity of microbes. And that's how a microbe works. If there's energy, I'll be there.
Reisenbach is especially interested in microbes from the Aquifocales family. filamentous bacteria that form dominant communities both in the hot springs of Yellowstone and in deep-sea hydrothermal vents.
The focus of my research is to identify them in a lot of these environments and then try and figure out what exactly are they doing in the environment, what energy sources are they using.
This member of Aquifocales is called Thermocrinus ruber. Millions of bacteria are packed together to form the visible biomass.
This big mass is not one single organism. There's a dominant organism, yes, that's the pink filament, but it's a community of organisms. The filaments that I work with at another site are covered in iron, just little black filaments. So they precipitate, take out of solution metals like iron, another case sulfur.
Members of the Aquifocales family are chemolithoautotrophs.
Chemolithotrophs use chemical energy that's associated with the hydrothermal fluids or with the hot spring water, and they use carbon dioxide. So they fix carbon dioxide and make organic carbon. And that's very similar to photosynthetic organisms. They fix carbon dioxide and make carbon, but they use, what kind of source, where do they get the energy source? Phototrophs.
Yeah, light energy.
Light energy.
In Yellowstone, photosynthetic organisms produce the top layers of microbial mats, tiny layered ecosystems teeming with life.
These oxygenic phototrophs, you see the oxygen here? That's the reason why we have oxygen on Earth today.
By understanding the metabolism of these microbes, scientists can begin to unravel the biological records left by similar organisms when the Earth was young.
Biological activity will eventually be incorporated into rocks, and so you can actually then begin to look back into the rock record and see when these organisms may have been active many billion years ago. So a lot of these fossils that are in these early rock records are long filamentous organisms, very similar to the kinds of things that we look here. They're also very similar to some cyanobacteria, so it's very hard to distinguish whether they are chemolithoautotrophs or whether they were photosynthetic organisms. And this controversy is still out there. We don't know what those original fossils were. Here you can see the edge of this mat that we obtained from Octopus Spring.
Back home at Portland State University, Reisenbach's work is just beginning. Her job? Identify the microbes she's captured from the park.
You look under the microscope and you see this community of organisms. You see a little blob and a little rod and a little blob and another little rod and a little blob. And so how do you distinguish between organisms that have no physical shape differences? And the only way we could do that until very recently was to try and grow those organisms from the environment. But just trying to second guess what these organisms like to eat just from their environment. Consequently, we've only been able to actually grow less than 1% of all the organisms you see under the microscope.
Using 16S ribosomal RNA technology, scientists can now identify organisms they could never grow in the lab. The tool is based on an evolutionarily conserved gene called 16S that all organisms share. The gene provides instructions for building a critical section of the ribosome. Scientists extract DNA from the samples and amplify out the 16S gene using polymerase chain reaction, or PCR.
You've got the 16S from this little rod bacterium, and you've got the 16S from this little round bacterium. You've got this little 16S from the little squiggly one, and they're all in the same tube. Now you have to separate out those genes, because what you want is you want to just get one of those genes, and you want to be able to sequence that information.
Fed into a computer, the new sequences are automatically aligned with the database of 16S genes.
For example, in this region, they're all extremely different. And so within the gene, this would be their diagnostic fingerprint. And so this now enables us to really identify organisms at the genus and species level based on this gene. This is for the first time in the history of microbiology, we're able to go into the environment and tell you that there's this huge diversity out there that has never been described before. What do you think they're doing? More than 90% of the diversity we see in the environment we've never described. And using these molecular tools, we can begin to describe that.
Finally, Reisenbach can generate a tree of life from the data. The computer shows her where the microbes fall, from deep on the trunk to farther out on the branches. Members of the Aquifocales family fall on one of the deepest and earliest branches on the tree of life.
Okay, so that would be the trunk of the tree. And so what you can see is that then the closest relatives of our universal ancestor to life would be organisms such as Thermocrinus ruber and these black filaments in Yellowstone.
These biological signatures are helping Reisenbach and colleagues establish the first public microbial database from the thermal springs of Yellowstone.
It's real important for the park to have such an inventory, because unless you know what you have, you cannot argue why somebody can't take something out of the park. Okay, and also because a lot of these microbes do have potential biotechnological significance. Pretty amazing, huh? Understanding what we have today, we will be much more responsible about managing our resources. Carl Woese once said, if you took away all the bison from Yellowstone, nothing will happen to the Earth as we know it. But if you took all the microbes away, this planet wouldn't be here. And so that also epitomizes that these microbes are the chemists of the world. And that's incredible.
New techniques in molecular science continue to reveal incredible diversity and surprises in the microbial world, including signs of communication between bacteria. Scientists have begun to decode a chemical microbial language, and they're learning that microbes aren't working alone. You encounter biofilms every day. It's the coating on your teeth in the morning, the slime on your contacts, that film in the toilet. It's the colorful scum around the hot springs in Yellowstone and the corroding scum on drainage pipes. At the Center for Biofilm Engineering at Montana State University, scientists are looking at bacteria in a whole new dimension.
Well, I think we used to think of bacteria as these very primitive, single-celled organisms with a limited capacity to differentiate, communicate, and cooperate. And really I think the most exciting idea in the biofilm field right now is that when these organisms get together in a biofilm, they act like a multicellular organism.
Biofilms can form on just about any wet and solid surface, which creates potential problems in medical settings.
Anytime that we put some foreign material in the human body, an artificial joint or a catheter or even a contact lens, there's a chance that bacteria will adhere to the surface and form a biofilm and cause an infection.
Biofilms also attach to living tissue. One of the best studied biofilms occurs when Pseudomonas aeruginosa colonizes in lung tissue in people with cystic fibrosis, causing chronic infections. Today, using a tool called laser scanning confocal microscopy, researchers can grow and study living biofilms.
What the machine does is it actually looks at one focal plane and then drops down to the next one, so you see a sequence of optical slices as it goes right down to the surface, which has been colonized by the bacteria. But then you can take that image and compile it and bring it back into a 3D image. Confocal's fantastic.
And it's opened the door to dissecting and interpreting a biofilm's life cycle. The first step, bacteria have to come in contact with a surface.
One can be a almost chemotaxis approach, where the organisms sense that there's an accumulation of nutrients on the surface, and they'll actually begin a random walk in that direction. Once they come in contact with the surface, they don't make an immediate permanent association with that surface. They may move around on that surface for a period of time, and once a certain time period has passed, they'll make an irreversible, and I'll say, decision to remain on that surface.
The bacteria that stay turn off the flagella-making genes and turn on the genes that produce the pili. The pili's twitching motility allows the bacteria to sniff out chemicals and aggregate on the surface.
They also start to produce the extracellular polymeric substances that act as that glue to stick them to the surface at that point.
We see signs of genes being turned on and off within 15 minutes of bacterium associating with the surface, so it's quick.
As the biofilm thickens, the bacteria make small signal molecules that wash over the other bacteria in the polymeric matrix. When the chemical messages reach a critical concentration, the bacteria start piling onto one another in diverse and dynamic structures. The ability to recognize and react to a crowd is called quorum sensing.
There's an awful lot of variation in what a biofilm looks like, from very thin, sparsely attached bacteria to structures that lift off the surface and look like a field of mushrooms or a city full of skyscrapers.
In the more complex structures, the microcolonies are separated by a network of water channels.
This could be a water channel here between a couple of clusters. There's another one up here.
So it's like a primitive circulatory system. It's bringing in nutrients, removing waste. It's been likened to the streets in a major city.
Researchers say that within 4 to 6 hours of first alighting on a surface, the bacteria begin expressing the new biofilm phenotype.
70% of the genes have changed, turned off or turned on. Now we have a whole new set of proteins, and a whole new set of gene products. So it's almost like the swimming bacteria, the planktonic ones, were the seeds, and now we're seeing the whole plant.
If you study an individual ant, you learn something about ants, but you miss the amazing things that groups of ants do together, the kind of communal behaviors they have. And I think that's the idea that we're coming to in biofilms.
But like anthills, biofilms are dynamic. If they get too large or parts of the biofilm don't get enough nutrients, some bacteria break away to form new biofilms.
And what we've noticed in Pseudomonas aeruginosa is it appears those organisms now start to express the flagellum again and downregulate the pili, and will actually evacuate certain portions of the biofilm and swim back off into the nutrient environment.
Infectious biofilms, like those in patients with cystic fibrosis, have a common denominator. They're all difficult to treat because of the diversity and specialization of the bacteria.
In a biofilm, there's some that see oxygen, there's some that see absolutely no oxygen at all. Some of them are growing fast, some of them are hardly growing at all. So you can't kill all of these classes of bacteria with the same antibiotic.
Our own immune system hits a roadblock with biofilms too.
Because single bacteria are about 1 micron in size, and a white cell is about 15 microns, so they can take them in. But if you get a biofilm that's about 100 microns in diameter, then it's a big cell like this, big mass like this, and the white cells come along and try to nibble away at the edges of it. And when they can't internalize it, they do a very nasty and invidious thing. They sit back and fire enzymes at it, like yay. And the bacteria in the biofilm couldn't care less about the enzymes, but the surrounding tissues get quite badly damaged.
Instead of targeting single planktonic cells, researchers are now studying biofilm-specific genes. The goal is to develop new strategies to fight infections caused by the biofilm phenotype.
So what I'm hoping to do with that is take the proteins, inoculate animals with them, and find out if they protect them from any kind of biofilm-mediated infections with Staphylococcus aureus. That's the first step. We can move on to human trials if we actually show any kind of efficacy.
New strategies include trying to disarm infectious biofilms at every stage in their life cycle.
And if quorum sensing turns out to be an important part of the development of a biofilm, then we ought to be able to cripple the biofilm or prevent it from forming simply by jamming that communication, disrupting the natural crosstalk that bacteria use as part of the regulation of forming a biofilm.
While some biofilms and bacteria are infectious, most microbes are not pathogenic. And because of their great abundance and diversity, they wield tremendous power. That's because the biosphere is driven by chemistry, and microbes maintain the balance. They are the recyclers and decomposers, unlocking carbon, nitrogen, and other elements essential to life. We continue to learn and benefit from these master chemists. While they're making a living, they're doing everything from cleaning up our environment to helping us be more efficient in industry. For centuries, humans have been mining the earth for precious metals. But in the process, millions of tons of low-grade ore are left behind. At the Idaho National Engineering and Environmental Laboratory, Dr. Frank Roberto is working with the mining industry to help recover these metals naturally.
Many of these metals are sulfide mineral-hosted metals. So in bioleaching, we use the microorganism which can utilize the iron and sulfur in the mineral to dissolve the rock matrix and liberate the metal that we're trying to recover in a mining operation. So bioleaching is basically biomining. So this is a clump of iron pyrite.
The liberating microbes are oxidizing bacteria, like Acidothiobacillus ferrooxidans, which won't grow unless the pH is very low.
Basically, you can think of it as battery acid. And it's using these inorganic substrates like iron and sulfur for its energy source.
Approximately 25% of the world's copper is leached biologically. Heap leach operations like this one operated by Kennecott can contain 1 million tons of ore piled on top a leak-proof pad. Adding water on top promotes the growth of the microbes and starts the bioleach.
This is a typical run-of-mine size chunk of rock. They break the rock apart, they produce acid, and one of the byproducts of sulfur oxidation is sulfate. So they produce sulfuric acid, which also helps to break the rock down. After the microorganisms have attacked the mineral and that water is passed through the heap, in a copper mine, for example, that water will be blue in color because of all the copper that's been dissolved out of the ore, and that then goes on for further processing at the mine site. This copper is about 97 to 98% pure. It can be further refined and leads to what we call copper wire.
A typical leach operation can take up to 2 years, and industry wants to expedite the process. But controlling bioleaching in the field requires understanding the microbial ecology of the environment.
In the heap, we have what we call microbial succession occurring as the temperature and other environmental parameters change.
To understand the process, Roberto is growing the microbes in a simulated leach operation.
And one of the things that we're still learning about is what is the full spectrum, the full dynamic of all these organisms? How do they interact? How do the populations change? What are the triggers? Is it only temperature? Is it nutrient source, water?
The goal is to stimulate different classes of organisms at different times.
Say we raise the temperature to 55 degrees, we would expect organisms like sulfobacillus. And if we raised it even higher, which is where our current interest is in trying to leach recalcitrant ores, say at about 70 degrees C, you would expect to find the Archaea like sulfolobus. What we're hoping to see is that these higher temperature organisms will be more efficient and we'll see more copper liberated from the mineral. Our vision of the future is one where biologists and miners work together, and basically the biology is another dial you tweak to get a better process.
But while the oxidizing bacteria are liberating the metals, they're creating new problems. Acid rock drainage seeping from abandoned mines is a major environmental problem impacting streams and groundwater across the western United States. At this abandoned gold mine in western Montana, pulverized waste rock left behind 80 years ago is polluting the environment.
Now, the reason nothing is growing on this site is that these mine tailings have too many metals in them and they generate too much acidity. So whenever rainfall falls on this site, it generates acidity, which prevents grasses and trees from growing.
To help reduce acid drainage, researchers are using sulfate-reducing bacteria. Sturman is looking for a low-cost organic carbon treatment, in this case cheese whey, that will facilitate their growth.
What we're trying to do is stimulate populations of bacteria within the columns here and within mine tailings on an actual site that are, number one, will help prevent the initial leaching of metals into solution, and number two, will then cause the metals that are in solution to precipitate out. So in a sense, the sulfate-reducing bacteria are reversing the reaction that the sulfur-oxidizing bacteria caused higher up in the column.
Sturman hopes his research will help accelerate the rehabilitation of sites like this.
So what we're trying to do is to take a technological solution and make a part of that solution the naturally occurring microorganisms that exist on this site and many other sites. We're just stimulating those organisms with a source of organic carbon that is in itself natural as well.
At another abandoned mine in southeastern Idaho, acid mine drainage has been polluting the environment since the 1940s. In a series of ponds and berms, the Bureau of Land Management is employing microbes to clean it up.
And we have pond 1 separated from pond 2 by this berm. And this berm is the heart of the treatment. It's called a bioreactor, and the bacteria are here.
The berms are composed of crushed limestone and a straw manure mixture. Sulfate-reducing bacteria live here naturally and work anaerobically as the water slowly percolates through the berm.
It was designed for a retention time of about 48 hours and seeps into pond 2. And this is where we expect the metals to come out of solution and deposit in the bed. And the bacteria are here working continuously to treat this water.
And the system is working. Treated water is being released into Champagne Creek just below the site, and it's helping restore the natural wetlands further downstream.
Right now it's having a huge impact, probably a 50 to 90% reduction on aluminum and copper and iron. We would like this system to improve the water quality of Champagne Creek over time, so much so that we can have fish migrate upstream in this reach, which haven't been here likely since mining started in the 1880s.
Microbes. With new molecular tools, scientists are just beginning to grasp the scope of their incredible diversity and chemical talents. They are profoundly changing how we think about the evolution of life and life beyond Earth.
We brought a rock back from Mars. What do we look for in the rock record? What are the signatures of life that we need to look for? On Mars, there's evidence for water. There was also evidence for some volcanic activity. And so the types of organisms that live in these hot springs may have existed on Mars many years ago.
In the field of biotechnology, however, we're trying to harness biology and biological products to do useful things. And there it's all about function, whether it's bioleaching of minerals, remediation of wastes. Function is the key.
The diversity of microbes is greater than the diversity of all plants and animals put together. We've only just started to scratch that surface of what is the diversity of the microbial world.
Funding for this program is provided by Annenberg CPB to advance excellent teaching. For information about this and other Annenberg CPB programs, call 1-800-LEARNER and visit us at www.learner.org.